Hey guys, I'm Tara.
And I'm Christian. Today we're going to be talking to you guys about receptors and signaling molecules, also some cell cycle checkpoints, and
then what happens if something goes wrong in the cell cycle? Cancer.
So beginning with signal molecules, we have two major classes of signals, the nonpolar ones and the polar ones. Basically, in terms of your nonpolar signals, those are going to be steroid molecules usually. Here we have testosterone pictured. You can see that even though it has these two polar groups, for the most part this molecule consists of carbon-hydrogen bonds, and so it's nonpolar. It can readily diffuse across the membrane. On the other hand, we have epinephrine, which has these four really spaced out and very polar groups that make it impossible for this molecule to diffuse across the membrane. And so for polar and peptide signal molecules, we have to have membrane receptors in order for the cell to respond to those signals. So we have two main classes of membrane receptors. That would be G-protein coupled receptors and enzyme-linked receptors. The G-protein coupled receptors differ from the enzyme-linked receptors in large part based on their production of second messengers, as you can see here. Every G-protein coupled receptor will make a second messenger, which then could activate a phosphorylation cascade, but those second messengers all will have different responses that they'll trigger. Enzyme-linked receptors, on the other hand, use these RAS proteins to trigger phosphorylation cascades. And so RAS proteins are a type of G protein, but you can see that they're not actually linked to the receptor the way that they were with the G protein coupled receptors. We need these bridging proteins to activate the RAS proteins once the receptor is bound by the signal molecule. And then the RAS protein is going to activate a phosphorylation cascade. So as mentioned previously, epinephrine is a polar signal molecule, and as such we need a membrane-bound receptor to receive the signal. So the beta-1 epinephrine receptor is a type of G-protein coupled receptor. As you can see, the second messenger that it produces is cyclic AMP, and you'll notice that the cyclic AMP will go on to activate a phosphorylation cascade, but there's no RAS protein to activate the phosphorylation cascade, and so that is how you would know that it's not an enzyme-linked receptor. So now we're going to move on to discuss the cell cycle, the checkpoints involved in the cell cycle. You can see there's 4 of them. Really we're going to focus on this G1 checkpoint because that's the one where if you have problems where the cell doesn't properly regulate the checkpoint components, you'll end up with cancers of various kinds. Beginning with a signal for the cell to grow, that signal will have the cell upregulate its levels of E2F and cyclin E. Cyclin E will bind to cyclin-dependent kinase to make this cyclin E CDK complex. This complex will act to phosphorylate the retinoblastonin component of the E2F, which this E2F-RB protein complex is the inactive form, and so in this form E2F will not be able to transcribe the proteins for DNA replication. However, once it's phosphorylated, you can see that the RB protein will dissociate from the E2F, leaving this active E2F behind and allowing for transcription of proteins. So if the cell realizes that the DNA is damaged, it's going to upregulate the levels of P53, which in turn will make P21 and inactivate this cyclin E-CDK complex. What that's going to do is it's going to inhibit the cell's ability to phosphorylate the E2F-RB protein complex, and thus the cell will never have active E2F and will not transcribe the proteins until we know that the DNA has been fixed.
So there are two different types of tumors. There's malignant and benign. Cancer tumors are considered malignant when benign tumors gain the ability to detach from their tissues and invade other tissues, which is bad news bears. So I have some notes up. Cells become cancerous when social controls fail in checkpoints. It's primarily seen in the G1 checkpoint. Cells can continue on with the cell cycle without the proper go-ahead signals, which is the main oncogenic mechanism. So the two types of defects seen when cancer is a result are when proteins required for cell growth are active when they're not supposed to be. These are upregulations of E2F and the cyclin protein, the proto-oncogens. And another defect is that there's prevention of tumor suppressor genes, and which shut down the cell cycle. These are P53, P21, and RB.